Okay, so welcome everybody to week 4. Today's topic is about disease ecology, and I'll explain a little bit about what that means. So unlike last week, where we talked about sort of populations and how populations have an impact on diseases, how diseases have an impact on populations, today's focus is really about the natural environment, and that includes things like ecology, biodiversity, as well as some other features of the environment, more physical features of the environment. So specifically today, the focus is on ecology, and so how different species relate to one another, how that has impacts on disease transmission. And then tomorrow we're focusing more on some of those abiotic factors. So landscape epidemiology, all about things like air, water, land, things like that. So to get us started today, how many bacteria do you guys think live in the average human body? You can just give me a guess what number. So a lot of different answers. So some of you are saying 3.9 trillion, 39 trillion, 3.9 billion. Okay, so diversity of responses here. Let's take a look. For those of you that said D, that was the correct answer. And full disclosure, I would never test you on something like this, but I think it is interesting to highlight just how many bacteria do live inside the body. And a lot of the focus of this week is going to be asking, well, why are some bacteria more harmful? What is it that makes some bacteria or some pathogens more harmful to us? How are we exposed to them? And what is that chain of infection? Why does it make some bacteria more deadly than others? So we'll try to answer some of those questions and more over the course of this week. So to get us started, what is disease ecology? Where does it fit in with all of these things? Well, as many of you probably know, given that a lot of you are environmental science students, ecology is the study of relationships between living organisms and their environment. Now, population ecology is the study of species populations and their interactions with that environment. So this is somewhat related to what we were talking about last week, right? Population dynamics, demography, but specifically population dynamics between species. So disease ecology takes some theories and some portions of all of that, and it's essentially the study of disease patterns, so how diseases are transmitted or spread over space and time, and how those actually have an impact on different populations of species. So we'll come to know some of these terminologies, things like host, pathogens, and all that. But the main thing to sort of distinguish disease ecology from these other fields, especially from something like epidemiology, which is the study of disease spread, disease ecology is really focused on those ecological factors. So what is it about ecology that specifically allows for the transmission of those diseases, as well as human environment interactions and then those pathogen host interactions. So what do I mean by pathogen? When I say pathogen, what is that? Well, it includes things like bacteria, viruses, other microorganisms like parasites. So anything that can actually cause or carry these infectious diseases. Now, a host is an organism that harbors the pathogen. So a lot of the time, especially today, we'll spend talking about human hosts, but as we know, many animals can also host an organism, host a pathogen. So it's not just humans that we would be worried about. Vectors as well are an important piece of this sort of disease ecology puzzle, and vectors are the agent which can carry or transmit the infectious pathogen. So an example of that, one that I think many of us are pretty familiar with, are mosquitoes. So mosquitoes are an example of a vector. And then a reservoir. Now, a reservoir is a habitat in which the agent, so whatever pathogen that we're talking about, normally lives in or grows in, multiplies in. So a reservoir can be an environment, right? It can be water, it can be air, but it can also be humans. It can also be animals, so other species. So a reservoir is a little bit more general. Now, One way to sort of think about this, because this figure here was, I think, the one that was in your reading. I prefer something more like this because I think it does sort of help us understand the relationship between these different things. Because in order to have disease transmission, you don't necessarily need a vector. A vector is a type of transmission. So some pathogens do use vectors in order to infect a host, but not all of them do. So I think this way is a little bit easier to sort of understand that the vector is not always present in that chain of infection. But something that you do need is this environment where the pathogen hosts, whether that's another host, a reservoir of some sort. As well, you need obviously the pathogen and then the host to infect. So those are the three sort of main components of disease ecology that you need in order to carry out that transmission of the pathogen. Now, pathogen types can actually get really, really complicated, and you can get really nitty gritty about classifying these different things. So full disclosure, you do not need to memorize this by any means. I just sort of want you to take a look at this and realize that there's a lot going on, and you can classify these quite in a detailed manner. But the main thing that we're going to focus on is that you can think about their modes of transmission as either being direct. So a pathogen will spread through something like physical contact, through direct contact with something like bodily fluids. So that's direct transmission. And then you can also have indirect. So that would be through vectors or through some environmental media. So it doesn't require that sort of human to human contact. So that's sort of the main thing that I want you to take away from a slide like this, is just that disease transmission can occur through those two sort of main types of transmission, direct and indirect. Now, as you might imagine, vectors also come into play here. So vectors, as we said, were a type of indirect transmission. Now, pathogens have different sort of needs and wants in terms of how they spread to hosts. So some of them prefer to use a direct mode of transmission. Some of them prefer to use indirect. Some of them prefer to use vectors. And they evolve in very different ways, these pathogens, in order to take advantage of a particular mode of transmission in order to infect the host that they want to infect. So what you, again, sort of need to know from a slide like this is that different pathogens do have these different preferences. These different modes of transmission don't necessarily work for every type of pathogen. They tend to really stick to one, especially once they evolve to recognize that one particular mode of transmission seems to really work for them in terms of being able to transmit to a host. So they'll usually stick to that. But different pathogens have those different preferences. So as you can see, viruses and bacteria tend to prefer the vector mode of transmission, whereas something like fungus, right? So spores tend to prefer direct contact, whereas helminths, which are a type of parasite, they tend to prefer indirect contact. So just something to know, something to be aware of as we go through these slides. Now, when we're talking about disease ecology, as you may have noticed, I'm not just talking about any disease, I'm specifically talking about infectious diseases. So diseases that are communicable, in other words, they're not chronic diseases, as we've talked a little bit about in this course so far. So they're communicable because they're caused by the pathogen spread. So those bacteria, those viruses, those parasites. But it is important to know that pathogens are only a risk to the host if they actually come into contact, right? So this is what the figure here is trying to show, that essentially, let's say you had a pathogen in some wildlife species, right? It only really poses a threat. It only has a risk of transmitting to humans if it enters this sort of domestic landscape where the human lives. And so that might happen through different modes. It might happen through infecting livestock, for example, if the wildlife comes into contact with livestock, might come into contact with other wildlife that does tend to live closer to humans. So there's many different ways for that to happen. But this transmission does have to occur within some sort of broader environmental setting, some sort of landscape where the human would actually come into contact with that pathogen, whatever it is and wherever the pathogen tends to reside. So pathogens do spread from animals, as we've talked about. So animal hosts or animal reservoirs, they can also spread through vectors or from other hosts. So human to human contact also happens with some pathogens. Now there's three sort of principles that I'm gonna talk about when I talk about disease ecology. So these are really the important main takeaways from a lecture like this. And I'm gonna go through them one by one, but I'll first sort of talk about them a little bit more generally. So the first principle of transmission of disease ecology is that the incidence and the patterns of disease reflect the interactions of populations, so of different species, within ecological communities, within an ecosystem. Now, diversity and density of different species is really important to consider. So diversity being how many different types of species are in an ecosystem, and the density being just that abundance of the species. So how many individuals in a particular species. The second principle is this idea of a chain of infection. And that occurs when a pathogen leaves its reservoir or some other host, and then through some mode of transmission, right? So either direct, indirect vector, it goes on to infect another host. Now, the third principle is infectious disease dynamics are impacted by overlaps in time and space of niches. And we'll talk a bit more about that in detail later towards the end of the lecture, but a niche is just a set of biotic or abiotic conditions in the environment that physically define or limit a species' ability to survive within a given area or within a given time. So I'm going to go through these in much more detail, so don't worry if they seem a little bit confusing right now as we go through it. So the first principle, as we said, it really has to do with species and the diversity and the density of those species. So the transmission of pathogens between species will be somewhat dictated by their distribution in an ecosystem and just their numbers, so their abundance of the organisms in an ecosystem. So as you can see in the slide here, when we're talking about individuals, those can form a population within the same species, but then a community can involve multiple different species, and all of that together forms an ecosystem. So most ecosystems will contain various distributions of different species. Some of the species will be more frequent than others, right? Some of them will have larger populations than others, and that is relevant to the abundance or the density of those species. So we said that diversity matters, but how? How does diversity of a host, for example, reduce disease transmission? What do I mean by that? Well, specifically, more diversity is equal to more non-susceptible hosts. So what I mean by that is that the more that you have a diversity of different hosts in a given ecosystem, the less likely a pathogen will find that one specific host that they can really thrive in. So it sort of dilutes the sample of potential hosts for a pathogen to really cause destruction in and cause an infection in. So the more diversity you have, the more non-susceptible hosts that might be there to dilute the transmission of the infection. More diversity also equals less encounters, because if you think about it, in an ecosystem, a lot of the time organisms are not actually bumping into one another. They're sort of actively avoiding one another due to predator-prey interactions. And so logically, the less that they encounter one another, the less contact they'll have with one another, and the less likely it'll be for that pathogen to spread to different organisms, to different species. But more diversity can increase the emergence of new pathogens. So even though we said that overall diversity reduces disease transmission, there is that one sort of exception that it can increase the emergence of new pathogens. And that's specifically due to something that we call a spillover event. So a spillover event just happens when a particular pathogen is able to jump species because they have contact with that particular species enough times, frequently enough. So that's a spillover event. What about vector diversity? So diversity of vector species also tends to increase the disease transmission. But how does that happen? Well, more diversity of vectors equals more ways to transmit the pathogen to a host, right? If you have a variety of different vectors available as a pathogen, you'll be more likely to be able to spread to that host. More diversity of vectors also means that you'll have more contact with multiple different susceptible hosts. So different vectors also have a preference to go towards specific hosts, right? A mosquito tends to target humans as well as some other species, but mosquitoes don't bite every single species out there. So the more diversity you have of vectors, the more likely you'll actually be able to spread to different types of hosts. So that's all that means. Now, what about pathogen diversity? So diversity of pathogens increases disease transmission. How does that happen? So more diversity of pathogens, the more likely you are as a pathogen to survive and evolve, right? That makes sense. If you have many, many different types of pathogens, the more likely that one of them will just happen to really thrive in a given environment and evolve to cause more infection in a population. The more diversity as well, the more resiliency you have as a pathogen to be able to thrive in a new ecosystem. So for example, if a pathogen is if there are many diverse different types of pathogens, then the more likely you will have one particular pathogen that has a real sort of knack for being airborne, for example, which then might allow it to thrive in a particular new ecosystem in a given environment. So as we've come to realize, the diversity of these different species is really important when we're talking about the transmission of pathogens. Now, I do want to point out, there's a reason I've put these arrows here. So the arrows here that I've added just means that the diversity of those different types also impact the diversity of one another, right? So the more hosts you have, the more likely that vectors will spread because they have more hosts to sort of feed on. So that in turn influences the diversity of vectors, and that in turn might influence the number or the diversity of pathogens that you have. So these things don't occur isolated from one another. They're all sort of interacting together, and the diversity of those different species will be really important in terms of their impact on the diversity of one another. So this brings us to the idea of natural selection. So as many of you might know something about Darwin, so natural selection is Darwin's theory where organisms that are better adapted to particular environments or particular ecosystems, those are the ones that tend to survive, and they tend to produce more offspring, which then allows them to pass on their genetic traits to the next generation. Now, as we started to talk a little bit last lecture when we talked about genetic drift and things like that, so we realized that pathogens can actually influence the genetic variety in a population, right? We realized that something like the Black Death actually influenced the genetic variation of people in England. So the English population to this day has a lower diversity of genetic traits than they did prior to the Black Death. So how do pathogens influence genetic variance in a population? Well, as we talked about, they cause illness, they cause death, and this, as you can imagine, would drive down the reproduction or the survival of those species. And so this affects the host's ability to pass on the genes, because if they die from an infection, they're probably not going to pass on those genes to the next generation. And so their genetic traits will become less frequent in the population. Now, we'll talk about this in the context of 3 different types of selection. So positive selection happens when you increase the frequency of those traits, those alleles. So for an example, a host might have particular traits that would allow them to have pathogen tolerance, right? So over time, positive selection would happen. Negative selection means that the traits that maybe didn't help the host survive eventually get eliminated from the population, or they become much less frequent because they are susceptible to the pathogen, right? So they don't tend to survive and they don't tend to pass on their genetic material to future generations. Now, balancing selection is something like a bit of a mix of the two. Balancing selection actually favors diversity. So they'll favor some positive selection genes, some negative selection genes. And I'll give you guys some examples so that that's a little bit more clear. So a malaria protective gene actually exists in a lot of populations in sub-Saharan Africa. Now, this is an example of positive selection because scientists believe that over time, the populations that tend to live in sub-Saharan Africa just naturally started to develop a higher prevalence, a higher frequency of these malaria protective genes, because obviously population that has those genes, right, that has more of those genes is much more likely to survive and pass on those genes to the next generations. So you can sort of see that in the figure here. So these are regions in sub-Saharan Africa where malaria is endemic. And the frequency of those malaria protective genes tends to be highest in many of those parts of Africa. Now, an example of negative selection is also present in the world in the Ganges River Delta. So this is a part of the world where cholera is particularly prevalent, being a bacterial pathogen. And because of that, low rates of blood type O is actually much more prevalent in that part of the world, in the Ganges River Delta, because over time, blood type O seems to be less susceptible to cholera. So it tended to provide them with, it was more susceptible, I should say, to cholera. And so this is an example of negative selection because over time, less people were surviving in the Ganges River Delta if they had the blood type O. So the less of them that were surviving, the less of them that were passing on their blood type to future generations. And that's why to this day, we see very, very low rates, the lowest in the entire world, in fact, of the blood type O in that particular region. Now, the reason that we have diversity of different blood types, scientists think, is because of balancing selection. So the reason we have A's, B's, and O's, scientists believe that different blood types with different sets of antigens actually provide human populations with different traits that allow them to be more tolerant to some diseases. And so they think that we have this diversity just to sort of keep the human population going, because in different geographic contexts, certain groups of human populations will tend to survive better with particular types of blood types. So hopefully that's clear. Are there any questions so far? If you would like me to spend a little bit more time on this stuff, let me know. Otherwise, I'll kind of keep chugging along. Okay, hopefully we're good. So this brings us to the idea of virulence, resistance, and pathogen evolution. So hosts cope with pathogen infections through resistance, right? They develop traits that can prevent infection. We saw that with the malaria trait, right? In sub-Saharan Africa, that would be classified as something like a resistance trait. On the other hand, hosts can also cope by evolving and having a set of tolerance traits. So these tolerance traits tend to reduce the impact of infection, right? So maybe they'll result in less deadly symptoms. Maybe the symptoms will be much more mild because particular genetic traits just happened to signal a tolerance in that host's body better. So they respond better to a particular type of infection. Now, for resistance traits, so something like the malaria resistant gene, let's say, eventually a risk of infection becomes so low in a population that these traits actually stop spreading. So the positive selection that we were talking about eventually stops. Why do you guys think that is? Any ideas? So why would something like a malaria-resistant gene eventually stop spreading within a population? No longer needed. They aren't necessary anymore if the disease isn't around as much. Yep, exactly. Yeah, so you guys got the right idea there. So for anyone who's kind of scratching their head, like, what does this all mean? Well, so resistance traits, think about it. If eventually the risk of infection becomes so low because a vast majority of the population, for example, has developed this resistance to a particular infection, they will stop having their advantage, their genetic advantage. And so eventually everyone keeps surviving. So the risk of infection becomes so low that it's not only the ones who've had the resistance trait who survive. Now everybody survives. And eventually this actually results in a sort of equilibrium in the population. And so it'll no longer be a case where just the ones with that resistance trait are prevalent. It'll actually be a very diverse genetic pool again. On the other hand, tolerance traits, they eventually result in a risk of infection being increased. And so these traits tend to continue to spread. Why do you guys think that might be? So kind of the opposite of what we were talking about with the resistance traits. Anyone want to summarize what might be happening there? Adapting to the disease's prevalence. Pathogens evolve to be more deadly. That's interesting. Yeah, theoretically, especially if it's something like a virus, which tends to evolve quite quickly. Any other ideas? Anyone wants to chime in? So for tolerance traits, eventually the risk of infection increases and these traits tend to continue. The main reason there being because they've got that advantage, right? So they will continue to pass on their genetic material because they're the ones surviving. And eventually, because they have such a strong genetic advantage, survival advantage because of these particular tolerance traits, you'll see the frequency of those traits just continue to increase over time in a given population. So that's why. This is particularly true with something like bacteria and parasites. So for example, in areas of South America, particularly in the Amazon, so around Peru, there's populations there that have developed an increased frequency of tolerance traits, specifically with soil helminths, so a type of parasitic disease. And that's exactly why, because those individuals who have the tolerance traits tended to survive better. And so they were the ones who kept passing on those genetic traits for tolerance to those particular parasites. And so that's why over time, the prevalence of those types of genes becomes much more frequent in a given population. So if the human genome is able to adapt to pathogens so well, why are diseases still so prevalent? Why are we constantly inundated with this sort of threat of pathogens around the world, resulting in infectious diseases? Well, as some of you said, pathogens do evolve, although usually pathogens evolve to become less virulent. This is because they actually want to keep spreading. And so if you think about it, if a given pathogen results in death right away, then it's less likely that that host will be able to interact with other hosts, right? Let's say it requires human-to-human contact. If a person is so sick that they can't get out of bed, then they won't encounter other humans, other potential hosts for the pathogen to spread to. So that's why, in order to survive, most pathogens usually evolve to become less threatening, less severe, less virulent over time. Now, another reason why diseases are still kind of constantly prevalent is because they infect new hosts, so they might jump to new species. And this is something we talked about a little bit with the spillover event. And as well, pathogens do develop resistance. So even if we develop some kind of treatment or some kind of medication, they do evolve, and so they develop resistance to a lot of those types of barriers that we might try to introduce, whether that's genetic or whether that's an antibiotic that we've developed. So unfortunately, despite the human genome's best efforts, pathogens are here to stay. Now let's talk about density. We haven't really talked about that. So now that we've covered the diversity of species, let's talk about density. So density of hosts and vectors, as you can imagine, relates to just their abundance in a given ecosystem over a period of time and space. Now, the diversity of host species impacts the density of hosts. What do I mean by that? Does anyone know why that might be? So if we have a greater diversity of host species, why might that impact the density of the host species? Any ideas? And it might help to sort of look at it from the context of these figures that I've put on the slide. So why would the diversity of hosts impact the density of those species? More diversity means there's more of them, so more dense. Species would want to stay in a particular environment, not spread due to predator-prey or carriers of pathogens in close contact. Interesting. This is such a range of different responses. Well, usually we would say that the more diversity of host species, someone else? Oh no, that's it. Okay. Let's look at these figures here. So in one figure, we've got only maybe half as many as people from the second figure here. And so their density is low, which means that you can have a bigger diversity of different people. So these things interact with one another in terms of when you have a diversity of host species, usually it reduces the density because there's less environment for them to cover. So for those of you that said it can increase density, it could theoretically, if you had this like unlimited environment for all these species to interact, right? But usually that's not the case. Usually they're constrained within a given environment due to the resources given, even things like temperature, right? Different species might only be able to survive in a particular small environment. So usually as you increase diversity, you actually decrease the density of the particular species because there's just less resources, less space for them to cohabitate in a given environment. Now, higher density of host species usually increases pathogen transmission, and it might be pretty obvious why. So if you look at this second figure here, it's just because the more density, the more abundance you have of a particular species, the more likely they're going to have encounters, the more likely that they'll have contact with one another and pathogens will be able to transmit that way. There are some exceptions to this, specifically with vector-borne diseases, because vector-borne diseases are more dependent on the abundance or the density of vectors, and they're less dependent on the density of hosts. So that's usually how vector-borne diseases are transmitted. They have less to do with the density of hosts and more to do with the density of vectors. So this brings us to that second principle of disease ecology, which has to do with the chain of infection. So this is when a pathogen leaves its reservoir, which can be, as you can see here, it can be a human host, it can be another animal host, it might be the environment. So these are all different types of reservoirs that are kind of like a home to that pathogen. And then through some mode of transmission, right? So we said that direct transmission can occur, indirect transmission, vectors are a type of transmission. Through this chain of infection, they will then go on to infect a susceptible host. So that's the chain of infection. Now let's look at an example of vector host transmission. So specifically here is West Nile virus. And vectors, as we said, are an example of indirect infectious disease transmission because they involve this sort of indirect intermediary to spread the pathogen from the original reservoir to the host. Now, some vectors are there to serve as purely a mechanical vector. So they're really just there as a transportation vehicle for the pathogen to kind of have a ride to infect that host. But other vectors actually play a really important role in the development of the pathogen. So an example of that would be something like malaria, because the parasite that results in malarial infection actually needs the mosquito's gut in order to come to maturity. And so the mosquito actually plays a core role in its kind of life cycle before it goes on to infect a human. So some vectors are much more important for that pathogen survival. Others just purely serve as a mechanical vehicle or transportation vehicle. So vectors include things like mosquitoes. We've talked about fleas, ticks are another well-known example. In this particular virus, so West Nile virus, the reservoir here is birds, and they develop West Nile virus. The pathogen tends to be more prevalent in certain bird species than others, and those mosquitoes will then bite the particular bird that is infected with West Nile virus, and they will transmit it to other hosts. Now, they transmit it to something called a dead end host. So both something like a horse and a human, in this case would be a dead end host, because West Nile virus doesn't have the capacity to transmit from host to host. It really only requires that vector transmission. So once a horse or once a human has West Nile virus, they can't infect other humans. So that's why it's called a dead end host. They would require that mosquito to then keep biting other humans or other horses in order for the virus to spread. Now, the burden of vector-borne diseases is pretty large, so they are by no means an insignificant part of the infectious disease story. Now, example of them include malaria, as I've said, Zika virus as well is a mosquito vector-borne disease, West Nile virus, Lyme disease, dengue fever, there's a whole bunch of them. So 17% of all infectious diseases in the world are vector-borne diseases, and they cause more than 700,000 deaths annually. So definitely an important piece of infectious disease. Now, where is the burden the biggest? Why are vectors dead end? Yeah, I can go back. So it's not the vectors that are dead end. Boy. So it's not the vectors that are dead end, it's the hosts. The hosts are dead end because they don't go on to infect other hosts. So in some infectious diseases, hosts can infect other hosts, but in vector-borne diseases, usually it's the vector that's transmitting the pathogen. So they are the ones that have to go on to keep biting hosts for the disease to spread in a population. So they're called dead end because they're not the ones that actually transmit that pathogen to other hosts. So hopefully that's more clear. Okay. So back to this. Where is the burden biggest of vector-borne diseases? This might be a little bit obvious, but I think it's worth pointing out. So where is the burden biggest? Africa. Yep. In general, what part of the world? Warmer climates. Yeah, that's also a really good observation. Although by that logic, Australia, you would think would be also with a significant burden near the equator. So what else? Less developed, yes. Poorer nations. So yeah, it's not just that it's climate, although of course climate does play a really, really important role because vectors are animals that require certain climatic conditions in order to survive. But specifically, it's really countries that have less capacity, less infrastructure to actually create barriers against those vectors. So in places like even Florida, right? Florida has a really hot climate that would arguably be pretty similar to some places in South America. But because of the infrastructure in place, right, they make sure to control things like mosquito populations. They frequently spray pesticides. Um, they also make sure to have the nets in windows, the screens, to make sure that mosquitoes don't come into people's homes. So those are examples of barriers that are pretty easy, pretty quick, but unfortunately they are not readily available in many parts of the world. And so those are the places that tend to be the hardest hit by these vector-borne diseases. Now, this brings us to Ebola. So Ebola is a zoonotic disease. Now, zoonotic means that the animal was the reservoir. So it wasn't some piece of the environment like water or air, it was specifically an animal. But not all zoonotic diseases are vector-borne diseases. So what I mean by that is that even though they may have originated in an animal, they might not actually need a vector to spread amongst human populations. So something like Ebola, although it originated most likely in a reservoir like bats, and then due to the pathogen's evolution, it was able to spill over into another species, and then humans likely had contact with that species through hunting or through gathering some animal products. And so it's no longer an example of vector-borne disease because the pathogen is spreading directly to humans, and then the human will go on to infect other humans, right? So it doesn't require something like a mosquito or a tick to carry on that chain of infection. The chain of infection is direct. It just requires physical contact. So this became the primary transmission route to infect other humans through other human-to-human contact. Now, for COVID-19, it's a bit of a mystery. Currently, we don't have a very good idea, but scientists' best guess is that it is a multi-host pathogen that likely also originated in bats as a reservoir, and then it was able to evolve through some other intermediate hosts, something like a pangolin, possibly. And then again, because the pathogen evolved, it was able to spill over and infect a human. So that's sort of their best guess. But time will tell. We don't actually really know. We don't have a firm grasp of what really happened there. Wasn't Ebola an epidemic in some countries a few years ago? Yes. So we don't unfortunately talk about Ebola very much anymore, but especially when I was doing my master's back in sort of 2014, Ebola was a really, really big deal. It was almost as big as COVID is now, but to a specific part of the world, right? So it was an epidemic in mostly West Africa. But it caused a huge burden of mortality, of loss of life there, specifically among healthcare workers, which is particularly unfortunate if you think about less developed nations who really rely on healthcare workers to do a lot of the work to keep populations safe and healthy. So unfortunately, yes, it is more deadly than COVID. Yeah. And it was very, very difficult for them to fight at the time, but you could say that because it was not endemic, but because it was so particular in one part of the region, there wasn't that sort of travel transmission that you would see with something like COVID, which likely originated in parts of the world where we were seeing a lot more international travel, right? There's just less people visiting West Africa. And so the transmission there likely didn't happen to spread into a pandemic. But yeah, you're right. Ebola was absolutely an epidemic. Now, why are some pathogens more dangerous in one host species than another, right? Why would something even like COVID not have necessarily decimated a bat population, but why did it cause so much trouble in humans? Well, as you might imagine, there's very, very different immunological processes that impact how a given host will respond to a pathogen. So for whatever reason, our immune system just seems to be much less tolerant to different pathogens. And scientists think that it's because humans as a whole are generally not as exposed to very many pathogens. And so unlike a bat, which has maybe had the evolutionary history to develop a lot of tolerant traits to different pathogens, humans just don't. Our genes maybe haven't evolved in that way. And that has resulted, unfortunately, to a set of immune responses, particularly in humans, that make it much more difficult for us to develop that tolerance or that resistance to certain pathogens. Now, why are some pathogens more dangerous for different individuals of the same species? So why do humans have a variation, right? Why do some people die from COVID and others not? In short, we don't really know that either. Among humans, of course, there's a whole vast amount of genetic diversity. Different genes might certainly play a factor, but there's other risk factors as well that we know influence disease tolerance. So whether or not someone will have mild symptoms or whether they'll die of a particular disease. So we know that immune responses might differ based on something like nutrition, even behaviors. So something like smoking, we know tends to decrease or suppress the immune system in ways that we know has an impact on infections. But even things like age, as we've seen, is a really, really important risk factor for some pathogens. So it's a little bit complicated, but this is, I think, going to be a really, really expanding area of research in terms of immunology and why some humans have certain traits that make them much more immunologically resistant towards some pathogens. Now, this brings us quickly in the last few minutes of class to the third principle of disease ecology. So infectious disease dynamics being particularly impacted by this idea of a niche. So remember that a niche is some biotic and abiotic conditions. So that means both the physical environment as well as the biological environments of species. So those environmental features define a species' ability to survive. So there's a reason why certain species only live in certain parts of the world. Now, this could also be said for something like a pathogen, something like Ebola, for example, we saw concentrated in many parts of West Africa for a particular reason, because they likely originated in the animals that, the reservoirs that were living in those nations in those parts of Africa. So how does this play out? Well, if we understand a human or a particular pathogen's niche, we understand where it's likely going to spread, right? So if something like a tick, in this example, is only found in a particular environment and it only bites a particular type of species, those niches are said to overlap. So we know that just because of the preferences of the environmental factors as well as the behaviors of the vector in this example, we know that that overlap might be the area where that risk of the pathogen transmission occurs. So a pathogen is really only a risk to human health when the human's niche overlaps with the pathogen's niche, either through direct environmental factors or through other species that serve as those intermediaries. So maybe it's a series of different overlapping niches of many different species. Now, I think I will end it there. I have a couple more slides, but I can save those for tomorrow. And they'll serve as a segue for tomorrow's lecture. So thanks, everybody. We'll continue with this tomorrow, and we'll talk about landscape epidemiology tomorrow as well. So we'll end it there.